Alzheimer's disease is a shadow that falls over millions of lives. It is a relentless illness of the brain. It slowly steals memories, thoughts, and a person's sense of self. Families watch as their loved ones drift away, even while still physically present. The emotional toll is immense. The financial toll is immense. It places a heavy burden on families and healthcare systems across the globe. This is not just a disease of old age. It is a profound public health crisis. The search for a cure, or even an effective treatment, is one of the most urgent scientific quests of our time. It is a quest fueled by desperation, hope, and billions of dollars in funding. This search for answers has been long and often frustrating. Governments, private foundations, and pharmaceutical companies pour vast sums of money into laboratories and clinical trials. They are all chasing a breakthrough. The central pillar of modern Alzheimer's research has been the amyloid hypothesis. For more than 30 years, this theory has provided a clear and compelling target. If they could stop the buildup of amyloid-beta plaques in the brain, they could stop the disease. The idea became almost an article of faith within the field. It guided nearly every major drug effort. Amyloid-focused research was more likely to be published in prestigious journals and to secure funding from the National Institutes of Health. This intense focus created a kind of tunnel vision. Alternatives struggled for attention and funding. Inflammation, metabolic problems, infections. Challenging the dominant theory was difficult. Dozens of anti-amyloid trials failed, one after another. Plaques were cleared, yet cognition did not meaningfully improve. Patients continued to decline. Troubling questions followed. Was the theory wrong or incomplete? Perhaps amyloid is a symptom, not the root cause. Or maybe treatments arrived too late in the disease process to help. One pivotal case centers on a landmark 2006 paper. Published in Nature, led by Sylvain Lesné at the University of Minnesota, it appeared to be a major breakthrough, identifying a species dubbed AV56 as a memory-impairing culprit in mice. It was cited over 2,300 times. Years later, physician-scientist Matthew Schrag spotted anomalies. Images appeared tampered with, duplicated. Formal reviews deemed manipulation credible. In July 2024, Nature retracted the 2006 paper. Misconduct allegations also reached within the NIH. A top NIA scientist faced scrutiny over dozens of papers. An internal investigation found image manipulation, including reused and altered figure panels. NIH moved to notify journals for corrective actions, including potential retractions. The scientists stepped down, raising oversight concerns. A third case involved a public company, Cassava Sciences, and a CUNY researcher, Huao Yan Wang. The consequences of flawed data ripple outwards. Flawed data, fraudulent data, causing damage far beyond a single lab. False foundations poison the well. Good faith researchers waste years and grants chasing a ghost. Fields veer down dead ends. Public trust is squandered. NIH invests over $3 billion a year in Alzheimer's research. Falsified grants steal taxpayer money. Unsound science drives costly failed trials. Patients and families bear the heaviest cost. These are not just a few bad apples. They reflect deeper systemic problems. Publish or perish pressures tilt incentives toward flashy positive results. Selective reporting creeps in. At worst, fabrication. Reproducibility falters when rigor is undervalued. Novelty outshines verification. Funding, publications, and prestige formed a self-reinforcing loop. Alternative ideas struggled to survive. Peer review seldom sees raw data. Investigations can be slow and opaque. In the wake of this scandal, the community is taking corrective action. First comes accountability. Journals are retracting fraudulent papers. Universities and the NIH are running internal investigations and making findings public. By withdrawing flawed research, we begin cleaning the poisoned well. Next, build a more transparent system. Mandate sharing of raw images, numbers, and records. Standardize automated image screening at journals. Publish what was actually observed in the lab. Agencies must adapt. Dedicate funds to replication studies. Back a diverse portfolio beyond amyloid. Resilience grows when monocultures fade. This reckoning need not be despair. Embrace transparency, rigor, and integrity. The path forward is difficult and necessary. Patients and families are counting on science to get it right.